Rebecca fait son doc University of New South Wales in Sydney en Australie. Elle vient d'Australie d'origine. Et par la suite, elle fait un postdoc avec Robert Coulter à Harvard Center pour l'Environnement. Et depuis combien des années? Quelques années. Elle est une professeure à l'University of Alberta en Edmonton. Et maintenant, elle prend du temps de passeport à Québec parce que sa famille est ici. Mais par contre, elle vient d'Australie, ça fait attention. Et elle a fait une année sabbatique en Singapour bientôt. Et par la suite, elle va revenir à l'University of Alberta. Je crois. Si. OK. OK. So, enfin qu'elle va faire la présentation en anglais, mais si on a un problème avec vous poser des questions en français, il y a des gens dans la salle qui peuvent traduire. OK. Parfait.
Is that on? Put that on here. So I'm from Sydney, Australia, which makes me sound a little odd in North America. But more importantly, I'm one of 5 children, which means I speak really fast to get in what I want to say because I'm competing with 6 other people. So while Americans do many things that are odd, something I loved and picked up during my postdoc is that they will interrupt when someone is giving a talk. So I strongly encourage you to just look at me quizzically or go like this and I'll stop and you can ask me anything. I love this data from the project, so I don't mind being interrupted at all. I've done this talk a few times and I strongly encourage you to do it. So it's particularly nice for me to be here today at Laval because I was thinking about it and it was actually in my early 20s that I came here for the first time. And I came because at the time I was having a lot of fun on student exchange in California, but I was starting to think about being an academic. And I think my parents thought I should get on with life. And so they paid for me to come to Quebec because my parents' best friend was on sabbatical at Laval. And sorry, iWatches come with problems. And so I got a plane over to New York. This was as cheap as I could do it. And then I got a series of buses up the coast to Quebec. And Godfrey and I spent several days walking across the plains of Abraham because they had an apartment just there, talking about what I was going to do. And then in his Irish way, in the end, he said, Beck, just get on and do it. So that is what I've done. And so for me, this is one of my favorite parts of the world. And I actually was supposed to go on to New York, but I stayed because he told me to travel north. And I went up to Tagisac, seeing whales and climbing through fjords. And I just loved it. It was like one of those moments in my life. And then later when I met my husband, who is Quebecois, I floored him because I came up to him and could pick that not only was his accent Quebecois, but he was not from Montreal. And that I had been to Northern Quebec and he grew up in Sept-Îles. So he was like, who is this person? So I had him intrigued. So this is one of my favorite places in the world. So I'm going to talk to you today about a project which I started actually during my postdoc, which was to look for small bioactive molecules, which we normally think of as antibiotics, but now this is really sort of expanding into the idea of hormones and any kind of small molecule that changes either the physiology or survivorship of the interacting species. And I'm going to talk about the molecules that come from this bacterium called Phaeobacter inhibens, which belongs to the Phaeobacters, which is a really common marine group. And they play a role in pathogenesis. And I'm going to talk to you a lot about an alternate mechanism of pathogenesis we've found, mediated by a secretion system, and how it activates apoptotic-like programmed cell death in Aemiliana huxleyi. So Aemiliana huxleyi is this unicellular phytoplankton, And it's considered a model species because it forms really dense populations over large areas of the ocean. We always joke, because there's a Belgian student in the lab, that blooms are bigger than Belgium, but they can be very, very large. And it's important because it makes two very climatically important compounds: dimethyl-sulfur-propionate that transforms to DMS to seed clouds, but also it's important in the carbon cycle. Not just because of photosynthesis, probably more importantly are these calcite discs, because that's mineralized carbon that can be deposited into the deep ocean for up to 50,000 years. So if we're thinking about carbon storage from phytoplankton, this is probably one of the best forms that we have. So this is the most numerically abundant form of the cell that's in the ocean, and it has 2 chloroplasts, so it's very small, it's just 5 microns, and it's a diploid cell type. We have 2 other cell types, the first being the haploid flagellated form that we can find in the environment, and we have just 1 culture in the lab. And this is actually the daughter cells, we think, of this calcifying strain, but meiosis has never been observed, and neither has syngamy. There's some debate as to whether it can accomplish syngamy in the literature, which is why it's a dashed line. Some people think it's fixed in its vegetative form. But these flagellated forms also have, like coccoliths, but they're carbohydrate discs on its surface. There's another cell type that was thought to be the same as this calcifying strain. It's also diploid, and it's the one most commonly used in the laboratory, because when you bring the calcifying strain into the laboratory, it rapidly transitions to this strain that has no coccoliths. And it was thought to just be a lab adaptation. But what we find is that you can actually find this cell type in the lab. Some really nice work done by De Vargas, and that it's not abundant, but it becomes more abundant at the termination of blooms. And that we find very important biological differences between these 3 cell types. So we use all of them in our group. There is just 2 azenic strains. Most people work on viruses with this strain, and so they don't worry about the bacteria. But you can only find these haploid and diploid at the moment in 2 single strains. We've tried to isolate, make them azenic, and we always produce these non-calcifying diploid strains. So the thing I'm really interested is in pathogenesis. And the reason I'm interested in is because I always think about how phytoplankton can die. And this is a process that I think is largely overlooked in the ocean, but extremely important because it's one of those big catalytic steps that happens in the microbial loop where we get dumping of biogeochemical intermediates in the ocean. And while we have constant rates of production and loss by processes such as primary consumption, because certainly Aemiliana huxleyi is food for grazers, they can also just become physically damaged or injured. So they're constantly then leaking metabolites. But we do see senescence, which is just aging. And we also see death by viral infection. This viral infection was first described by Woody Wilson, but Kay Biddle went on to describe really nicely programmed cell death, which he thought at first was apoptosis in Aemiliana huxleyi. And it's the first case of where they've really suggested that there's programmed cell death in unicellular organisms, because this was really thought to be a metazoan, a multicellular animal process. Okay. So what I think has been largely overlooked is the potential for bacteria to be pathogens. So while this is just 5 microns, and I certainly get reviewer comments, which is this is just too small to be a host, we actually see them coded in bacteria. We see many more bacteria than viruses in live samples. And you can live very healthily with bacteria coating the surface, and the bacteria we work on all directly attached to their host. So we think this is a direct, not a passive sort of molecular diffusion interaction that's going on. So I started working on a variety of phytoplankton when I first started my postdoc. And the reason was I spent 2.5 years in my PhD trying to grow juvenile red algae in the lab. And I promised myself that I would never make another graduate student do that. And so I got really interested in mainly strains that were producing this DMSP because of its sort of interesting climate role. It's also a chemotactic signal, so bacteria will sense it and swim towards it, like they swim towards amino acids or sugars. The bacteria really, really like it. And so this is the strain we sort of got working well in laboratory assays because I really wanted to have something more tractable than just working in the field, because I had done that during my PhD and I wanted something I could transition from the environment to the laboratory. And so we sort of established this model during my postdoc for how we thought bacteria interacted. They will rapidly colonize from an extremely low density to Ameliana huxleyi within 24 hours. And we were able to detect this compound, which we were surprised about, fumaric acid, we call PCA. And the reason we were surprised is this is a derivative of lignin, which if you want to ask me about lignin, I'm happy to talk about it, but I won't talk about it today. But it is actually produced when terrestrial plants are damaged or aging. So we consider it like a senescent cue that it's made when EHUX ages. And when we supplied the bacteria with this metabolite we could get from phytoplankton, they would be stimulated to make actually a whole lot of different small molecules. But one of the most abundant ones were this novel class of antibiotics, which are active against algae, which we called rosibacticide because they came from a rosibacter. And they can kill at nanomolar concentrations, which is extremely efficient, better than the amoxicillin you might take if you get sick. So they're very, very potent. But we had done all of this with purified compound. And so as I started my lab, I really wanted to start the experiments, which was what happens when you're doing organisms, not molecules and organisms. And so we got those 3 cell types in the lab and were able to colonize them and set up reproducible experiments, which doesn't take no small amount of time. But we got this very unexpected result where we saw that actually the cell type, the bald one that was susceptible to the rosibacticides, was not killed, and the other 2 cell types were killed. So what you're looking at in the graphs in the bottom panel is that over time, 2 weeks, we would co-culture both the bacterium and the phytoplankton together. And here you can see we're measuring maximum quantum yield, which is a measurement of the function of photosystem 2, which for a phytoplankton is essential. And so this is a really good proxy for health, and we find it to be one of our most responsive measurements. And so in black, you're seeing the co-culture, and the control of EHUX by itself is in the white. So what you can see is it maintains quite a high maximum quantum yield over the time of the experiment, but you can see this very rapid decline in photosystem health in the co-culture for both the coccolith producing strain and the flagellated form as well. So we wanted to really make sure that this was really happening, that that we were seeing death of these other 2 cell types, but not death of the cell type that we had shown with the molecule. And so what we did was a series of experiments with purified molecule for these 2 cell types. And we know that the LC50, which is the measurement we make for what is sort of like a toxic amount of the compound at 0.19 micromolars. And then we did it 10 times higher and 10 times lower, because drugs often actually have different therapeutic uses in different concentration ranges. That's why they can, you know, be used for a cold in one situation and used to grow hair in another situation. That's where a lot of drug development goes now. So what we could see when we did this with the different rosibacticides for the different strains is that we got the same fluorescence or chlorophyll reading for all of this. Lean and Anna did this so many times under so many conditions, but what we decided in the end is we took senescent or aging ones and just were saying, if you're really unhealthy, can I kill you with this molecule? And they just can't kill them. So we thought maybe it's something to do with the delivery of the molecule. We need the sort of mutants in the pathway to see if we have a bacterium and we mutate the pathway, maybe then we would be able to make it survive with the mutants. So what you're looking at here is in the control is just Aemiliana huxleyi by itself. And then because all the mutants require kanamycin to be maintained, we add kanamycin and we have a little effect, but not much of an effect on chlorophyll fluorescence. And then we have mutants at all different parts of the pathway towards the production of this rosibacticide antibiotic. And you can see that all of them still are able to kill. So we can definitely conclude that this killing mechanism is actually a rosibacticide independent process. And the exact same can be said for the flagellated form. And so at this stage, we knew, okay, it's a pathogen and it has multiple mechanisms of pathogenesis, not just the production of antibiotics or these antibiotics, but we really didn't know what they were. And so we sort of concluded this work and we thought, okay, we're going to switch over to the host, because at the time we didn't have a way to necessarily move forward with trying to work out with molecular biology how this was actually killing. And so what we decided to do is to start to look at the host, because a lot of work had been done out of Kay Biddle's group and then Vardy's group to be able to describe how viruses are able to use sort of viral glycosphingolipids, these lipids, to actually induce the death of Aemiliana huxleyi. And as I had said, that Kay Biddle had shown, I thought first it was apoptosis, but then Vardy's group has shown really nicely, it's actually this independent process called autophagy. And the distinction here is that autophagy allows the recycling of nutrients into the viral particles. And this made a lot more sense because the viral particles are requiring all of their nutrients because they're intracellular within Aemiliana huxleyi. And so it made sense that they would use autophagy to kill their host. And so how they do this is that the viruses get incorporated and then produce these glycosphingolipids that are able to insert themselves in their host membrane. And this disruption to the membrane actually creates the activation of proteases that are cause caspase-like activity. And the reason we call them this is because caspases are required in apoptosis, but they can be used in other programmed cell death pathways such as autophagy. And so this led to the autophagous programmed cell death of Aemiliana huxleyi. What they were able to see is what we saw, which is this rapid decline in photosystem 2 health and the loss of chlorophyll. And so I've already shown you that. But they were also able to see the caspase-like activity as well as a spike in reactive oxygen species. And so these are all signatures of apoptosis, but also specific to autophagy and a few different types of programmed cell death as well. So I'm going to tell you just a little bit about how this caspase activity works, because it's essential to understand how this assay works to understand the work. So caspases are in the cell and they're proteases, and they're normally in their inactive form. But for them to become active, they actually need to be cleaved. And how they get cleaved so they have the right folding is through stimulation either of reactive oxygen species or the viral glycosphingolipids. And so Aemiliana huxleyi can do both with the viruses. And what's really neat about this is the caspases recognize specific little protein sequences. So they're not proteases that just degrade everything in the cell. They'll specifically go to cellular structures required for life and break those down. So it's a real, we really think of it as a programmed cell death because it's specific. But what's also neat about them is that you have lots of them, and once one is activated, it will then go over and cleave another one, and it creates a cascade. So you're getting more and more proteases over time, which become more and more active, which is why you get that rapid decline in death, because it's basically amping up the killing process. It's not something that happens linear, it happens exponentially. So what it looks like is you have this activated, like Pac-Man-like protease, and it will come up and cleave the target protein, which will have its specific peptide. And then as it does that, will produce apoptotic-like programmed cell death. And we can supply a fluorescently labeled substrate that when it's cleaved becomes fluorescent. So when you're seeing green fluorescence, that means you're getting caspase activity because it's digesting that substrate and making it fluorescent. What really distinguishes autophagy and other programmed cell death processes from apoptotic-like programmed cell death is that when you add a specific inhibitor to inhibit the caspases, it will stop that cascade and you'll actually get recovery and you'll get no death. So while ever you still get sort of an alteration in photosynthetic health, you might get some ROS. Once the caspases are inhibited, that will quieten down. The cell will deal with those disruptions, repair, and then recover. And this is what Biddle couldn't show for the viral infection, is that when they added the inhibitor, it slowed death, but it didn't recover from the death at all. So what we were really suggesting is that bacteria would come and stimulate caspase-like activity, and that if we truly have apoptotic-like death, we would get what I've already shown is this loss of chlorophyll and chloroplast function. We would also see an increase in reactive oxygen species. We'd see caspase activity. So this is just like viral infection. But what would distinguish it is that we could add the inhibitor and it would shut down the cascade. And that we would also see this additional process where specifically the caspases will activate the cleavage of DNA. So you'll start to get DNA being cleaved, and the nucleus will deform and you'll get blebbing of the nucleus, and then eventually you'll lose nuclear integrity and DNA degradation. So that's what I'm going to show you now. So we started with experiments where what you're looking at is that control in white, which is healthy, it's EHARCS by itself. And then in orange, you have the co-culture where you have this sustained period of health and then this rapid decline. And we took samples from early time points where there was no disruption. They're living happily together, but not having an apparent effect on each other's health. And we added a similar fluorescent substrate for reactive oxygen species. So if you look along this axis, it's forward scatter. This is a proxy for size. And this is the fluorescence for reactive oxygen species. So if we set up gates here so that below that gate, we say that 95% of cells are below. And so we consider that not fluorescent cells, so they don't have elevated ROS. Of course, all cells have ROS, so you have to set some kind of threshold for what is an allowable amount of ROS. And we said, so if it goes above that line, we're getting an increase in fluorescence. So the fluorescent dye for ROS is happening. And then we set also a threshold for size so that 95% of cells are in that quadrant. I know it doesn't look like it, but it's just a dot plot. So if they become smaller than that, we're really thinking of them as dying. And so we can see that if we get this shift in the population. So what you're looking at here is the time series I showed you before, and the control is in black, and in orange is the co-culture. And you can see that at day 4, there doesn't look like any elevated ROS, perhaps a little by day 6, but clearly by day 8, and it's very pronounced by day 10, you can see that most of the population has elevated ROS within the cells. And day 12, there still is, but it's starting to decline. And that's because cells are pretty much dead. So that's really when actually ROS generation stops. And you'll see by day 14, we create this subpopulation of cells that are smaller, and they're breaking away because they're dying. And so that's what's going on. So we can see this under the microscope as well. And so what you're looking at here is just a control EHARCS. Some EHARCS have heaps of coccoliths. This one just has a single layer of coccoliths on the outside of the cell. That's the roughness. Bacterial people always think it's a biofilm. It's not. It's just coccoliths. And then the chlorophyll in the chloroplast has red fluorescence. But you'll see that we are not staining at all for any of the caspase activity. However, if we take it from the co-culture, you'll see that it dramatically looks different. You'll see that some of the cells have actually lost chlorophyll fluorescence, but all of the cells are staining for caspase activity. Anyone who is interested in coccolithophores will immediately notice that there's heaps of coccoliths on the outside and they're not arranged properly. That is something we're super interested in and pursuing because it seems like the bacteria can alter coccolithogenesis inside of the cell, but I do not have a story there for you yet. But we can quantify it over the entire population. And you can see this large induction of the caspase activity on day 10 in the population, and that's what that normally looks like. What it is here is just the days, and then you measure the relative activity of cleaving the peptide IETD over the entire population. But then really importantly, Anna did the experiments looking at adding the caspase inhibitor. So for the 10th time, you're seeing the same experiment where you have healthy EHUX in the white and then co-cultured with Phaeobacter inhibens with a rapid decline. If we add just the inhibitor to the control EHUX, you see there's no influence at all, and that's what you expect. It's just a little protein inhibitor. It shouldn't affect the cell. But if we add it with EHUX, I was actually surprised this worked so well and made Anna do it many times. You can see that there's complete recovery of Aemiliana huxleyi, and this is different to viral infection where there's only partial recovery for a little time and then you get complete killing. So this indeed starts to really look like a true apoptotic programmed cell death inside of EHUX, and that program is reserved for bacterial pathogens, not viral pathogens. But we wanted to obviously look at cell numbers as well, because really we're not just interested in photosystem 2 health, we're interested in the survivorship of the cells. And so you can see that the EHUX population is quite dynamic over time, just growing by itself. You add in bacteria and you can see that sharp decline. But if we add the inhibitor, it doesn't change the population at all of control cells. And indeed, when you add the inhibitor, you get survivorship of cells and they have that strong photosynthetic health. So these are indeed not just healthy cells, these are cells of the same population density as well. What was interesting to us is throughout these experiments, we always track the population of Phaeobacter inhibens, the bacteria as well. And you can see that Phaeobacter inhibens grown by itself just in the seawater medium actually can go from 100 cells per mil to 10 to the 5 cells per mil. And then it sort of declines a little bit in starvation. You add EHUX and you get a sort of one-fold increase in the cell density. And that makes sense because EHUX is a phototroph. It's going to leak organic carbon and nutrients, which the bacterium is going to be able to live off. And so that's quite a large population increase, right? It's 1,000% increase in the population. But over time, you can see that it continues to increase. And so we counted when we add the inhibitor, and we actually get a lower cell density of the bacterium. And we attribute this to that rapid death of EHUX is actually creating a massive nutrient pulse for the bacterium that it's able to live off. And this explains why the bacterium would activate this programmed cell death inside of EHUX, is that it increases its cell density almost tenfold. And that is a massive increase in population density, and strongly would ecologically explain why they would kill their host, because people ask all the time, why would a bacterium kill its host? Well, if its host is dying, producing senescent signals, and becomes compromised, its metabolic rate is slower, and it will start to sink out through the water column. These bacteria that I work on are specialized to live with hosts, both animal and algal hosts. And so they are adapted to living in that photic zone environment and want to stay there. And so they certainly don't want to be trapped on their host, sinking down deep into the ocean, because their attachment is actually irreversible. There is a Quebecois professor who works on this little protein he calls the Spider-Man protein. And this protein attaches just a specific group of bacteria related to Rosebacters to cells. It includes things like Phaeobacter. And when I saw him speak for the first time, I said to him, oh, my cells look like that. And we looked And indeed, this bacterium has a homologue of that Spider-Man protein. And so with that irreversible attachment, unless they kill their host and it breaks up, they wouldn't be able to then produce a flagella and swim away and become pelagic again. So the next thing we needed to really show, besides the inhibition of this caspase activity and caspase cascade, is the effect that it has on DNA inside of the cell. And so if we look at the control cells, when they're grown by themselves, they have a very clear, small, dense nucleus. However, with these infected cells with Phaeobacter inhibens, you can see that the one that still has chlorophyll has a nuclear area, but it's not as tight. And those that have no chlorophyll anymore and have activation of caspase have DNA throughout the cell. And that's indicative that the nuclear membrane has been degraded and the DNA is through the cell. So certainly DNases will start acting on that. We can also see the characteristic nuclear blebbing you see in humans and Drosophila and our model apoptotic organisms. So what you're looking at here is the nucleus of Emiliana huxleyi when it's infected with Phaeobacter inhibens. So normally a nucleus will just be small and round and by itself. But what looks like two nucleuses next to each other is actually a single nucleus, and that the membrane has become distorted. And so that's really characteristic of apoptosis. And when we look on flow cytometry, where we're using forward scatter and then staining it with cyber green, which stains specifically DNA, you'll see the control in red forms a discrete population, but in the back, the black smear are all those cells that have a declining and mixed amount of DNA. And that's characteristic of cells undergoing apoptosis because their DNA is cleaved and degraded within the cell. And so as that happens, it's a dynamic process. Some have lost more than others. So from this, we can really say that Phaeobacter inhibens activates apoptosis or an apoptotic-like programmed cell death within Emiliana huxleyi. And we're pretty happy at this point, but we still didn't know how Phaeobacter inhibens was doing it because it wasn't using its algicidal antibiotic to do it. And so we decided to do some comparative genomics work because there wasn't genetic tools in Phaeobacter at the time. But there was an increasing number of Phaeobacter genomes available. And so I had been working on this strain that is from Spain, and it was isolated from coastal waters. But some work done out of Brinkoff and Thomas's group had sequenced a strain 210 that I'd been part of isolating in Sydney from Ulva australis. And what was surprising was how similar they were, even though they were from different sides of the earth. So they had the same gene order. The gene order in the entire genome is completely conserved, and they have somewhere between 88 to 93% protein coding conservation. But what is different between them is a couple of specific things, inserts into this pathogen's genome. We also know from the natural product chemists that they have the same production of characterized secondary metabolites. So they both make rosibacticides, this algicide. They both make this very potent antibiotic and anti-cancer compound called tropodiphytic acid. They both make the plant hormone indolic acidic acid. They make the same siderophores, and they also make something that's algicidal and a siderophore called rosiaquilin. So Mohammed Sayada Sayadamost, who identified the rosiaquilins, did the rosibacticides with me during my postdoc, and I saw him 2 days ago in Boston, and he says he has another antibiotic from them, but he hasn't named it yet. So there's certainly a third, and he said both of the strains make them. So we know that their activity profile is the same. So we decided, because we could isolate it down to a couple of things in the genome, that we would co-culture these two, as well as a variety of different ones I won't show you. And you'll see that EHUX by itself is perfectly healthy. You can infect it and kill it with our strain we've been using. But when we use this Australian strain, it doesn't kill it at all. And I made them reculture it and check it over and over again, check that it didn't lose its plasmids because it has things on plasmids. And this is for real that it didn't. So at this point, while we knew all the antibiotics or bioactives were the same, we knew from the genome that our strain that was a pathogen had both a prophage and a type 4 secretion system encoded in it. So we're pretty sure it was one of these two things. And it was literally within weeks that, and this is why it's very important to make friends as grad students and postdocs, someone working at JGI, Rex Malmstrom, who I'd known from my postdoc, sent me a letter and said, Beck, this is on my desk. They're making a massive transposon mutant library of your Phaeobacter strain. Should we work on this? Yes, 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 yes. So they made the large transposon mutant library, and we got first pick of the mutants that we wanted to work on. We did a bit of work with the prophages, but there's really a problem expressing it with both the group that described it, Brinkoff, and then we worked with Matt Sullivan. We can't reliably get anything out. So we've really focused down on this type 4 secretion system. But to talk about that, I'm just going to tell you a little bit about Agrobacterium tumefaciens, which is like an intro micro model organism. So most people know it. But it was really important to me because actually Rosibacters were only named in the early 90s, which doesn't seem that long ago to me. And until that time, they were called marine Agrobacterium. They're pretty closely related, but they are distinct clades. But what's different about them is Agrobacterium is a dedicated soil pathogen. And it's a different type of pathogen in that it doesn't kill its host. It's more like a parasite, whereas ours fully kills the host. But it also uses that same signal, the chimeric acid, and it uses that as a signal because Agrobacterium tumefaciens only infects slightly damaged plants. So either when they're aging or if there's a wound, it will produce a breakdown product of lignin, and that will signal the bacterium to infect. And then once it does that, it absolutely requires its type 4 secretion to infect and produce these cancer-like growths, which we call galls on plants. And so how it does that is you can find Agrobacterium in soil, but you most commonly find it in the rhizosphere immediately around the roots. It's able to orientate itself and get towards the plant, just like Rosibacters. And it will associate with these roots. And then once it senses that it's damaged, it's able to attach to the plant. And then from a plasmid, it encodes this secretion system. And the secretion system spans from the bacterium to the eukaryote. So it can transport things directly into the cytoplasm of the eukaryote host. And once it does that, it actually has this little, we call oncogenic region of DNA, and a little endonuclease cleaves the plasmid and binds the DNA. And then this DNA protein complex will move into eukaryote cell, and it encodes for it to produce the cancer and make metabolites for Agrobacterium tumefaciens. This is how we have transgenic plants. It's the only way we sort of know, well, it was one of the early methods for being able to do genetics in plants. So it's pretty important, but it's not just that DNA complex that goes through. Most people know that. There's a bunch of small proteins that go through it as well. So this is what it looks like. What you will see is that there's an inner and outer membrane of the bacteria, and it's a very sort of complex structure with lots of proteins that coordinate to make an apparatus through the two membranes, and then it makes a syringe-like structure that will able to pierce the eukaryote host cell. And it has some specificity for these proteins here. So the people who had done the genome had found a couple of the big genes, and we went in and were able to find the entire operon. And most of the whole structure is encoded in this VERD-D in this direction, the purple, and the multicolored VERD-B in this direction. And that makes the big structure for this. And we could see this in the Phaeobacter inhibens genome. So I asked Rex for everything he had, and he had a bunch of different mutants, and these are transposon mutants. So sometimes they disrupt, and then it can disrupt the operon. So we wanted as many as we possibly could to be certain of what we were doing. So how we did these experiments is in the same way. We have our control, we have our wild type, but now we also add kanamycin, because to maintain the mutation, you have to have kanamycin there. And the first one we chose was a gene at the very beginning of the operon, the lytic transglossaryl. It's actually an orthologous replacement to VERB1 in the classical operon. And indeed, we saw complete attenuation of virulence. So what this told us, which is that like Agrobacterium tumefaciens, it completely requires this secretion system to be able to infect and kill its host. And so then we went on with all the structural components. There are 2 ATPases, as well as something that's between the bacterial membranes, and that sensing protein on the end to make it specific for eukaryotes. And all of them produce the same result, that if you mutate them, you get complete recovery of the host, in that it's unable to infect and kill. Just before we'd done this, some work had been published in eLife by Segev saying that it wasn't rosibacticides, it was indolic acetic acid that was the active molecule. And so indolic acetic acid is this plant hormone. We'd worked on it a bit in cell-to-cell signaling, and we really didn't think it was that molecule. And it's never known to go through a secretion system. So we wanted to check all of our mutants. And what this basically tells you is it's not indolic acetic acid. So we have our avirulent strain 210. It's the highest producer of indolic acetic acid, and it doesn't kill. But then we have our strain that does kill. It makes indolic acetic acid. And when we look at all of our mutants, we get some change in the production, but we never lose production at all. And it's within actually a small error for chemistry. Even when we add tryptophan, the precursor for indolic acetic acid, it doesn't change the stimulation of the molecule at all. So we can conclusively say that plant growth hormone is not how this is being killed, but we don't actually know what's going through the type 4 secretion to kill Aemiliana huxleyi and activate that apoptosis. So we went back to the operon, and the reason we did that is because in Agrobacterium tumefaciens, you have these 2 operons, the VERB and VERD, and they make that structure of the type 4 secretion. But in between are all your effector proteins. So you have your VERD2 that cleaves the DNA, binds it, transfers it, and allows the incorporation of bacterial DNA into plant genomes. But you also have these D and E units that are transferred, and they actually are critical to virulence as well. And so what we were able to find is this little light blue area, or aqua area, we've called VERU, and it was originally annotated as 2 hypothetical genes. And we went in and could see that we thought there was at least 3 genes and perhaps a gene fusion with a gene going another direction, but we haven't shown that yet, but at least bioinformatically, that's what it looks like is going. And fortunately for us, we could also detect 2 domains. So these are not the whole proteins, they're still hypothetical genes, but they contain 2 domains important to eukaryote ubiquination. And so, or ubiquitin sort of transfer. And the student I had, she actually had started with Antarctica fieldwork and then had done the apoptosis, which I thought was a stretch. And so at this point she was like, Beck, I just no idea what this is. And this was my second happy dance because this definitely is a mechanism that things like salmonella that give you foodborne illness, it does something quite similar to this. And so what these domains E1 and E2 do in ubiquination, I'll describe in a minute, but actually the first thing we did was the experiment because we had it all set up. So we threw it in, and again, complete attenuation of virulence. So this makes it a really good candidate effector that's going through the type 4 secretion and actually doing the death, causing the death. So how would it do that? Well, ubiquitin is this very small sort of peptide that is in eukaryote cells, and it will bind to an E1 domain, which is what I was talking about, and then it can get transferred to an E2 domain that will then anchor to the protein E3 that holds a protein. And the E3 would be present in the Emiliana huxleyi host. It's there. What this then allows it to do is E2 transfers the ubiquitin onto the eukaryotic protein, and it just keeps doing that to produce this, what we call polyubiquinated tail on the eukaryote. As it does that, we're able to, the proteasome mechanism, the proteasome apparatus is able to recognize the ubiquitin tagged protein, and then it's passed rapidly through the proteasome, and actually the ubiquitin is recycled and the amino acids are as well. And so this would provide the catalytic activation of the caspases, because they require some kind of proteasome activation. So what our working model is that Phaeobacter inhibens as a bacteria will sense EHUX and come and colonize it. And then once it does that, it produces a type 4 secretion system that spans the bacterial eukaryotic membrane, allowing it to deliver in its effector proteins directly. Once it does that, this produces ubiquitin cleavage inside of the host, which activates caspases, producing that apoptotic-like programmed cell death. So we're now sort of focused on trying to either get the sort of proteomics work to identify which proteins are going to be ubiquinated, or whether we can isolate the specific caspases that are ubiquinated. And we think that is probably the next step. So with that, I have to give so much credit to the people who work in my group. We are a marine lab in the middle of the prairies. And so most of them come from the coast and are very dedicated to come from the coast to live in Alberta to do this work. Particularly, I need to mention here Anna Bramucci, who did the apoptosis work on a lot of the co-culturing, and Lene Lebau, who is the other PhD student who started all of this work. All the chemistry, the early chemistry, I actually should have Mo on this slide, but it's not really part of the talk. But the later chemistry we do with Tilman Harder's group in Bremen, and a lot of and also Pauline Della Matta at the U of A. And Fabini Orata is the bioinformatician that we work on in this work. And especially I have to mention Rex Malmstrom and Elizabeth Ryan, who works at him at JGI, for providing all of the mutants for us.
Thank you very much. That's fascinating. Is there any questions?
You guys did not interrupt. I was like, you're going too fast.
No worry. I'll go ahead.
Yeah.
Yeah, I think we were just controlling the outcome. So, well, thanks a lot. That's really interesting. And of course, it's such a fascinating model because Vidiana is a major player in the world ocean. So your results raise a lot of very interesting questions about how the ocean works. I had a couple of questions. One was, in many of your graphs, you're showing no effect until 7 to 10 days, and there's some catastrophic cascade. What's happening during that period of time? Is there some critical threshold, the inhibitor, or is it a matter of attachment of the bacteria?
So we know that the bacterial cell density definitely gets to like its maximum stationary phase, and EHUX does as well. We certainly see that EHUX actually has to start declining a little bit, and I think that is the senescence. So we got really caught for a long time. It was rosibacticide because all the physiology looked like the senescent cue was important. Maybe it still is for these guys. Certainly when you add chimeric acid, you get a shift in the proteome. So it's affecting more than just rosibacticide biosynthesis. So there's those physiologies that are going on. We also know that there's quite a bit of interaction that happens during that time. While there's no direct benefit to EHUX's health, because we use a medium that's supplemented with B12, but if you make it oxytrophic for B12, I'm sure there would be a benefit because EHUX doesn't make its own B12 and Phaeobacter can make the B12, so you could produce that. But what one master student has shown, I didn't show that today, but he's worked on the tetrodotoxic acid, the antibiotic that it makes, and our idea, and it's very common at the moment in microbiome studies, is that this idea that bacteria provide this chemical defense and protection to their hosts. And so we wanted to look at, because we think it goes through a symbiosis and transitions to pathogenesis. And so indeed, it does work quite well in providing a chemical defense, but it's really specific. If you add them both at the same time, the alternate pathogen that will immediately kill the host, so like a more traditional pathogen, it will just outcompete the antibiotic producer. The antibiotic has no effect. You can change cell density. That doesn't matter. It will still outcompete. What you have to do is make the alternate pathogen a secondary colonizer. So if you play with succession. So if you have Phaeobacter inhibens as your primary colonizer, so it's there, then it can chemically defend subsequent colonization, but it cannot outcompete pathogens for initial colonization, which I would suggest that's probably, in terms of bioactive molecules, that's what's going on, but probably the B12 is what's happening in terms of survivorship for metabolism. I think they're the two basis of the symbiotic phage.
So what is happening at day 7, day 10? You're finally passing that.
Yeah, at day 10, what you're seeing is the bacteria have been at their highest concentration. EHUX is starting to decline, so it's senescent. And then it's set off, yeah, and then it does. And you can, because we purposely start with younger, more logarithmic growing cells and with a low bacterial concentration. We thought if we could just add more bacteria, then it would kill earlier. No, you can add 10 to the 6 cells per mil, and it will still have that delay until EHUX is ready to be infected or killed.
Yeah, Roger?
I'm just curious to know, I noticed briefly you mentioned about plasmids.
Yeah.
I just checked when you were talking, and I know there's something like 4 plasmids in some of these isolates. Is there any link of these plasmids to some of the phenotypes you're looking at? For example, do they encode some of those inhibitors or some of those antibacterials, or do they regulate something by transcription? Because I'm interested in that very much, because we've been looking at plasmids in Pseudomonas aeruginosa, but you can't find plasmids homologous to a whole collection of environmental isolates.
Yeah.
So I'm just curious to know, do you know if there are any ink groups from these things? Do we know anything about them?
Yes, so there's definitely plasmids amongst this group. In our strain specifically, and one of the things we're really we liked it because of this, working on type 4, is that the type 4 is in the genome. In other strains, it's in a plasmid, and that can just be much harder to work with. So we like that. The antibiotic tribodecytic acid, which some of those enzymes are also used in Rosebacter biosynthesis, they're on plasmid. That's actually been one of the big roadblocks. When I was a grad student, I had a few practicum students from Germany, 2 of them, and they both spent a lot of time either curing plasmids or trying to get plasmids in, because plasmid incompatibility was a big problem with getting genetics to work for the strain. Interestingly, I got interested in this when I spent a brief time in Halifax in Ford Doolittle's lab during my PhD, and I was just there learning bioinformatics, and I started looking at plasmids in Rosios because it was still when a genome was a big deal and a plasmid was what you sequence first. And that was the first time I saw an AT plasmid inside of a Rosebacter, and that's when I started thinking, oh, is this related to Agrobacterium? So it's one of the things I would love to know is about plasmid compatibility between these groups, because they seem to be able to handle TI and AT plasmids. So, yeah.
Must be a tough cookie working with Doolittle.
With what?
Working with Doolittle, that must have been something.
Oh.
Because I know him very well.
Yeah. So here's a complete pleasure. The thing I always tell this story that I think is hilarious is we were going, both when you were going to lab meeting, he would never let us all go in the same lift, because he was worried about our safety, but mainly worried that the whole lab would disappear in a lift crash. But the other time we all went hiking for 3 days, and apparently there was a postdoc who stayed behind. He was in the lab 24/7, didn't sleep because he was so worried the whole lab would get eaten by bears or die or something. It's very anxious about stuff like that. But he's amazing. Like, he's such a pleasure.
I noticed some of these isolates come from so-called not very cold waters, but
yes.
If we go into the Arctic, when you have these algae, do you have this bacterial species and these algae, are they there?
Yeah, so we've looked at this a bit. 16S is problematic, which a lot of the mapping, so definitely like that group, we get sequences pretty much everywhere. We probably get a bit less in certain areas, but I know that it's like Lorna Graham has isolated it down in Antarctica when she was in a cruise, tribotidic acid producing ones. She has the genome, but I think he's working on it and hasn't made it publicly available. Torsten Brinkhoff as well has done isolations, but mainly for other roseos. He was working on kelps and stuff, and so he got other ones out. These are distinguishable because they're really dark brown because of the antibiotic. It's brown in color, so people tend to pick them off. There's not a specific culturing protocol. My husband works on Vibrio cholera, and I forever hate him because he has Vibrio cholera plates. I would do anything to have a medium that just was my bacterium. So it's a bit hit and miss. It's really when you're specifically looking for it. Of course, I can't tell you, I've made so many classes at Banfield, culture these things. I find all kinds of Pseudo-Ultramonas. I'm yet to find Phaeobacter in Banfield. So. Oh, it grows on heaps of media. It's just everything else does. So to find it is just, you know, yeah. My students now say they give me 4 weeks while we teach out in Banfield, and they'll teach the lab doing it, and we get through a few thousand that way. But I've said I'll pay for a semester if people just go culture it, but no one wants that project. Yeah.
Yeah.
Thank you for your talk. You showed us what happens to a new unit when you infect either with bacteria or the virus. Have you tried infecting with both? Assuming in nature.
Yeah. So you sound like someone in my lab. They all want to do this, and and then I say, but how will we get the viruses out of the lab and then trust our results afterwards? So definitely, I want to, and we also really want to add a grazer in because the grazer increases DMS and DMSP production, which is the chemical cue for it. So it'd be great to get to those trophic levels. Also, because now we have the mutants in those pathways, you could get to which molecule changes which ratio and food webs. Yeah, we just haven't got there yet. Yeah.
Amazing how much there is to do.
Yeah. And not that many people do grazing on EHUX. There's a couple, but yeah.
Yeah. What would you use as a grazer?
I'm just trying to think. I know that because I joke with Alistair about it, his cafeteria strain can definitely eat it. But there's another one which is more common in the environment. I'm just forgetting its name, but they've used that as well. But the cafeteria one.
Okay. Est-ce qu'il y a des autres questions? Okay, thank you very much.
Thank you.